Hi, and welcome to poster 458. I'm Matt, and I'm going to spend the next 5 minutes walking through how the Breath Biopsy VOC Atlas can be used to contextualize putative candidate VOC biomarkers across different disease settings. For those who might be new to the breath field, the field is focused on the measurement of volatile organic compounds, or VOCs, in the breath of patients. These VOCs originate from metabolic or biochemical processes in the patient and can either rise systemically, diffusing from the target organ into the blood, across the alveolar membrane into the lungs, and then exhaled on breath, or locally within the airways themselves. A significant challenge, though, in breathomics is understanding the fundamental biochemistry underlying some of these candidate VOC biomarkers, and therefore contextualizing how the biomarkers might be used downstream in a downstream clinical pathway. The Breath Biopsy VOC Atlas is a curated reference data set of VOCs collected from a representative population, with VOCs listed as either on or off breath in that population, allowing us to start to contextualize some of these VOCs in the wider setting and understand the clinical use cases for some of these biomarkers. For the work in hand, we set out to undergo a comprehensive literature review to identify potential VOC biomarkers in asthma based on previous literature publications. And we did this agnostic to collection or analytical approach. We then contextualized these VOCs identified from the literature as discriminatory in asthma with the Breath Biopsy VOC Atlas to serve as that reference data set. This atlas contains a representative population of around 94 people, including healthy and disease-affected individuals, with samples collected via the receiver breath sampler and analysed via thermal desorption, gas chromatography, mass spectrometry. We categorise these compounds in the atlas as either on or off breath, with on-breath compounds being those that were elevated at least twofold higher than the background in paired samples and detected in greater than 10 samples across the atlas cohort. We use this classification and apply this classification to the literature review to flag those literature review related VOCs as either Pathophysiology-Associated, i.e. those which were identified in asthma and on breath in the atlas, or Pathophysiology-Specific, i.e. those identified in asthma and not on breath in the atlas. Our Prisma formatted search resulted in 21 papers which met all of our inclusion criteria. From those 21 papers, we identified 225 unique VOCs as potentially discriminatory between asthmatic and non-asthmatic individuals, with 47 appearing in multiple papers across the field. Those belong to a wide class of chemical classes as flagged in figure 2b. We then compared these 225 unique VOCs to the Breath Biopsy VOC Atlas, with 46 VOCs overlapping between the literature review and the atlas. 36 of these were on breath in the atlas, so classified as Pathophysiology-Associated, and 10 off breath in the atlas, so classified as Pathophysiology-Specific. We then dug a little deeper and started to contextualize these VOCs based on that criteria. Namely, for our Pathophysiology-Specific biomarkers, we've taken here nonane and octane as examples. We can understand from the literature these are associated with lipid peroxidation and inflammation, a setting we expect to be elevated in the airways of asthmatic individuals, but potentially not present at baseline in a representative data set, explaining why they may be pathophysiology-specific in this setting. For acetone and furfural, examples are pathophysiology-associated biomarkers. These are associated instead with fatty acid metabolism or dietary ingestion, phenomena we'd expect to be present at baseline, so on breath in the atlas, as we see, but potentially altered in a disease setting, such as asthma. We therefore walked through how the Breath Biopsy VOC Atlas can be used to contextualize VOC candidate biomarkers found in literature, but also internal study settings. We flagged how these can be correlated to potential biological interpretation of these compounds and contribute to understanding of biomarker relevance as well. It is important, of course, to note that in an emerging field like the breath field, this could be due to differences in collection or analytical methodologies as well. And it is expected that as the field expands and starts to harmonize, this will decrease moving forwards. We're excited to be increasing the Breath Biopsy VOC Atlas with additional data sets comprising healthy and diseased individuals, and this is now freely available for academic users and can be accessed using the QR code on the bottom right.